Hello, welcome to Nerve Physiology presented by the SHRTAB. We reference Colored Laws of Physiology by Agamemnon de Bostoulos and Stefan C. Bernard. Okay, this is the nerve system and the muscular physiology. So we start with the nervous physiology. So we start with the structure of the neuron. So the structure of the neurons, you are going to have the cell body and you have the axon terminal. So this is the cell body here made up of the soma, and here soma just means body, and then we have the dendrites here. So the dendrites here, many dendrites coming at this position show that this cell is going to be a multipolar neuron. At this position here, you have the axon hillock. At the axon hillock, you have the action potential that is going to be produced. So we have at this position here of the dendrites, you have multiple generator potentials which are being produced here, and then at the axon hillock at this position here, you have an action potential that is going to be produced. So now the next position here, you have the axon at this position here, you have the axolemma here, you have the endoneuron here. So now at this the different grooves that you have at this position that you can see here are called the knots of Ranviers, and here all through the neuron myelinating like that is going to be the myelin sheath, and this myelin sheath is going to prevent the loss of conduction. So now you also have collateral neurons. Collateral neurons can be functioning in reducing the nerve impulsing or increasing the nerve impulsing. And here you have the presynaptic ending or the terminal button. So at the position where you are going to have neurotransmitters. So all this, you're going to visualize all these positions, all these parts in the next part of the tutorials. So here we have the electrical transmission. So when the electrical transmission come at the presynaptic ending, so when the electrical transmission come at this position here, we have the binding. We have the cause of that electrical transmission is going to cause to the binding to the membranes, and that binding is going to cause now the release of the neurotransmitters. So now the next position here, we have this passive ion transmission. The passive ion distribution is going to be seen here. Then you have the active sodium ion potential pump. Sodium ion, you see that 3 sodium ions is going to be pumped normally outside, then 2 potassium ions are going to be pumped outside. But we know that at the rest, the cell are permeable to potassium ion only. So the potassium ion can also move outside, and they are going to produce a resting membrane potential of negative 90 millivolts. So we also have the closings. The external part is going to be much more concentrated. Is going to have much more positive ions compared to the internal part. You see that chloride ion is going to move outside, also outside of the cell. So since the chloride ion move outside, it's going to produce again more of the negative resting membrane potential. So that's why in total you have the resting membrane potential of negative 90 millivolts. So now let's visualize now the equilibrium potential. I've initially told you from the cell physiology that we have the resting membrane potential or the membrane potential could be found from the Nernst equation. And now if you substitute each of these values of potassium ion and then sodium ion, calcium ion, hydrogen ion, chloride ion, hydrogen carbonate with their respective concentration, as you can see here, you are going to have these respective equilibrium potentials. So you are going to have this resting membrane potential, all this with respect to these ions. So now let's come back now to the action potential of a nerve. So the action potential of the nerve starts, so it has to respect this diagram. So it starts with the resting membrane potential at the rest. Then we have depolarization. Depolarization occurs when you have, so what occurs at depolarization is when you have excess conductivity of sodium ion, as you can see in this other diagram. So you have depolarization, which is phase 1. It occurs when you have the conductivity to sodium ion, that is sodium ion is moving inside the cell. So when sodium ion is moving inside the cell, since sodium ion is positive, it's going to render the cell more positive, and that's why the membrane potential is going to tend toward a positive value, and that's why it's going to be plus 30 millivolts as its maximum value here. Then since excess sodium ion has moved inside, now you have the next portion, which is phase 2, repolarization. Is going to be there's going to be increased conductivity of potassium ion. So potassium is going to move outside the cell. So when potassium ion move outside the cell, is going to, since potassium is positive, is going to reduce the positivity inside the cell, and that's why the graph now is going to move downward toward the negative side. And now there is going to be this part which is going to call hyperpolarization, because the gates of the potassium ion channel have a reluctance to close. Since there is this reluctance to close, it's going to cause excessive repolarization of this membrane, which is called hyperpolarization. And then you have now the this last phase where the sodium ion potassium ion pump are going to take place again. So now these are the different parts of the action potential. So now let's visualize the voltage gated sodium ion channel. So this is the closed phase of voltage gated sodium ion channel. When you have a stimulus, it's going to open itself, and now you have different toxins that are going to cause the prevention of the opening of the sodium ion channel, which is called tetrodotoxin and veratridine. So all of these are going to prevent it from closing, tetrodotoxin. So now we have, normally when sodium ion enters, it's going to now be inactivated again later during repolarization. The prevention of this inactivation is via veratridine and batrachotoxin. So these are the points that you visualize here. So now let's see now the continuous and the saltatory propagation of action potential. So you see that you need to know that there are certain type of neurons that have saltatory propagation and other have continuous propagation. A saltatory propagation is going to be given via this myelin sheet. So now when you have myelin sheet covering the cell, you need to know that that's going to produce a saltatory propagation. And now when there is no myelin sheet, it's going to produce a continuous propagation. So you need to know that what at this position there's going to be stimulus. So when there is stimulus, it becomes positive here and then negative. This this stimulus, when you have stimulus, it's going to cause now this position inside to be positive and this position outside to be negative. Now there's going to be conduction to the next portion, as you can see here. So it's going to cause this part to be positive and this part to be negative. The next conduction again is going to cause again this other part to be positive and this other part to be negative. So that's how action potential is being transmitted through the membrane. Action potential is just defined as the rate of depolarization, or the rate of the depolarization of all these channel proteins. So when these channel proteins are being depolarized much more, that's going to produce what is called action potential transmission. So now we have, because this is a continuous propagation, you see that with continuous propagation, actually there's going to be a reduction in amplitude with respect to time. Why in saltatory propagation, where you have this knot of Ranvier separated by myelin sheet, you have a stable amplitude with respect to time. So as you can see in this diagram, you see that the amplitude is going to be smaller inside the myelin sheet, but when it comes to the next knot of Ranvier, the amplitude is going to be great again. So this is why saltatory continuous propagation is going to be faster and is going to be much more conservative. This is why saltatory propagation is going to be faster and much more conservative than a continuous propagation. So now let's visualize now what occurs at the level of the synapse. So this sodium ion is going to enter, you have action potential. So now the action potential is going to cause now the calcium ion channel to open. So the calcium ion, the opening of the calcium ion is going to cause ion channel is going to cause calcium ion to enter the cell. The calcium ion which enters the cell now is going to bind to the cal modulin, and which is going to cause the activation of the calcium ion kinase 2. Also, the calcium ion main role is going to cause the binding to the synaptotamines, and the synaptotamines are going to bind to these vesicles, these stable vesicles containing the neurotransmitters. The stable vesicles now containing neurotransmitters are going to be converted to unstable vesicles, which are going to bind to the cell membrane and cause the release of the neurotransmitters. The neurotransmitters which are being released now are going to bind to the different receptors on the postsynaptic membrane, and these receptors could be called: number 1, ionotropic receptors; number 2, metabotropic receptors; and number 3, cation channels. So these are the different parts. So these are the different conductivity diagram that you can visualize at each point. At this terminal, which is before you arrive the synapse, you have the action potential. At the synapse, you have this potential, a negative potential, and then you have transmitter release. The transmitter release, you have this, which are with respect to the concentration and time. So now let's see what happens next. So when you have the binding of the sodium ion, the binding of sodium ion of the neurotransmitters could be on ionotrophic receptor or metabotropic receptors. So the question is, what are ionotrophic receptors? Ionotrophic receptors are receptors that when you have the when you have the neurotransmitters binding to those receptors, it is going to cause the direct entry of the ions. The chemotransmitters binding the neurotransmitter binding to those receptors is going to cause a direct entry of the ions. While in metabotropic receptors, you have chemotransmitter binding to a primary receptor, and that primary receptor is going to cause a secondary messenger that is going to bind again, that is going to produce a secondary messenger that is going to bind to another cation channel, and that's going to cause now the movement of the ion. So as you can see here, you have this is going to be called the electric potential stimulating. So it is called the EPS1. These are all the multiple summation of stimulation that we have of action potential. It can come at different times, as you're going to see in the next section. So now let's visualize now what is called spatial summation and what is called temporal summation. So before that, let's visualize this name. This name is excitatory postsynaptic potential. The EPSP is excitatory postsynaptic potential. So now this first diagram is going to represent what is called a spatial summation, and this next diagram represents what is called a temporal summation of the stimuli. So now let's visualize what is called a spatial summation of the stimuli. With spatial summation, you need to know that the different stimulus are occurring at the same time at different dendrites. So we have one dendrite here which has an action potential, another dendrite here has an action potential, another dendrite here has an action potential. So this action potential, when it enters the new neuron, it is now going to be called an excitatory postsynaptic potential. So it is no more the action potential. You need to know that, as I initially told you, an action potential is produced as from the axon hillock. So in this new cell, you have no action potential, but instead the generator potential or the excitatory postsynaptic potential. So now you have this action potential coming from the latter cell, the presynaptic cell, and then it comes now producing the excitatory postsynaptic potential. But now one excitatory postsynaptic potential is unfavorable in producing the action potential, because this action, it is at negative. You need to know that what for an excitation of the action potential, or one action potential, you require at least a negative 50 millivolts. You require at least a negative 55 millivolts. Now this first action potential AP1 has a maximum of negative 70 millivolts. So only AP1 is not effective in stimulating the action potential of the postsynaptic cell. So what is going to occur is that in spatial summation, you have the EPS1, the excitation potential of the first cell is going to add up excitation potential of the next cell, which is going to add up with the excitation potential of the next cell, and all of them are going to produce a new action potential of negative 30 millivolt, and negative 30 millivolt is higher than negative 50 millivolt, which is the minimal voltage required to produce an action potential at the axon hillock. So now when you have now that stimulation is going to produce the action potential now from the axon hillock towards the axon, that is going to be called the spatial summation. Now in temporal summation, you have only one excitatory postsynaptic stimulus. So or you can have two, but most of the time it is one. But now at this time it comes at different times. So for temporal summation, you have stimulus coming at different time. So now this stimulus has a voltage of negative 70 millivolt, and this other stimulus have a voltage of negative 70 millivolt. But now the time difference is, you have a certain time difference between the two voltages, between the first excitatory postsynaptic potential and the second excitatory postsynaptic potential. Since you have that time difference, the time difference is going to sum up partially to produce a slightly above negative 50 millivolt, which is the minimal voltage to produce an action potential in the postsynaptic cell. And that, since it is higher, is going to cause now action potential in this next cell. So now this is what is called now an inhibitory postsynaptic potential. The inhibitory postsynaptic potential, this is an excitatory postsynaptic potential. Here you are going to have the depolarization. When the neurotransmitter enters, you can have sodium ion that is going to move inside. When sodium ion move inside, it's going to cause an excitatory postsynaptic potential. For an excitatory postsynaptic potential, you are required to increase the amplitude of the amplitude of that potential. The amplitude of that potential is supposed to be high, such that it is going to be above negative 50 millivolt, which is the minimal voltage required to simulate an action potential. And now you have an inhibitory transmitter. For an inhibitory transmitter, you see that the binding of the neurotransmitter is going to cause the movement of the potassium out of the cell. This movement of potassium out of the cell is going to cause now the reduction of the membrane potential. The reduction of the membrane potential is going to prevent now the activation of the action potential. You see here that the excitatory postsynaptic potential goes at negative 70 millivolt, but the inhibitory postsynaptic potential goes below negative 90 millivolt. The summation of this is going to produce now a lower membrane potential, which is going to be now inhibiting an action potential in the postsynaptic cell. So now let's see down the next one, the termination of action potential. So now, how does an action potential terminate, or what is the fate of the neurotransmitter? So now we need to know that the neurotransmitter bind at this level. Some of them could be reabsorbed and could be re-encircled inside vesicles, can be re-entangled inside vesicles to be stored again. So that's the first fate. The second one is that it can bind again to its own cells, so that are autoceptors. So that's the second one. So we have the first one, reuptake. The second one, it can bind to the own cells, called autoceptors. The next one we have again, and that autoceptor now is going to prevent, now this autoceptor is going to prevent again the binding of calcium, is going to prevent the binding of the vesicles to the presynaptic membrane. So some of them can bind to the autoceptors, which are going to prevent the binding of the vesicle to the presynaptic membranes. The next one, we have this enzyme, the enzyme breakup. So the first enzyme breakup is this. We need to know that monoamino oxidase. Monoamino oxidase is used in cases of catecholamines, when you are speaking about acetylcholine, and when you are speaking about adrenaline and noradrenaline. So the enzymatic breakup is going to be via monoamino oxidase. So this enzymatic breakup can be occurring as a next phase. The next phase is that some of them can bind to the receptors, and these receptors can be loose, can be broken down, and then you have diffusion out of the cleft, and you can also have, it can enter into the glial cells. So all these are all the fate of involved in the termination of the neurotransmitters action. So now let's visualize all the neurotransmitters found inside the nervous system. So this is a transmitter. We have the receptor subtype. We have the receptor type. If it's either ionotrophic is going to be represented in orange, and metabotrophic is represented in green. Then we have ion conductance, which ion is mainly involved in these receptors. And then we have the secondary messenger. Secondary messenger are only involved in cases of metabotrophic receptors. So now we first start with acetylcholine. Acetylcholine, we have nicotine. So acetylcholine, there are two main receptors which are involved in neurotransmitter acetylcholine. It can be nicotinic receptor, and it can be muscarinic receptors. Muscarinic receptors can be divided into M1, M2, and M3. Nicotinic receptors are what? They are ionotrophic receptors, and these ionotrophic receptors can have both an increase in conductance of sodium ion, that is, sodium ion enters the cell. It also have an increase in conductance of potassium ion and increase in conductance of calcium ion for nicotinic receptors. Then we have muscarinic receptors, which are M1. So M1 are metabotrophic receptors, and they function via the activation of the IP3, that is inositol 1,2,1,4,5 triphosphate and diacylglycerol. So M1 function by the activation of IP3 and diacylglycerol. Then we have M2 and M3. M2 and M3 function as a metabotrophic receptors, and it only function with increasing in potassium ion channel conductance, and it function also and the secondary messenger involved here is going to be an inhibition of the adenylate cyclase, that is prevention of CMP production. Then we have ADH. So vasopressin is V1 receptor, V2 receptor. We can have also CCK, which can go from A to B. So all the vasopressin are going to be V1 and V2 are going to be, the receptors are going to be metabotrophic. We have this V1 is going to be via the activation of this one, and this is via activation of CMP. Then CCA, we have is going to be only metabotrophic and is occurring via the activation of IP3 and DAG. You have dopamine. Dopamine, we have D1, D2, and D5. D1 and D5 are both metabotrophic, and they are going to be via the activation of CMP. While D2 is going to be metabotrophic and goes via the inactivation of the CAMP. So since he has an increase in conductance of potassium, you see that D2 is mostly involved in inhibition, and also M2 and M3 are also involved in inhibition. So the more you have a potassium increase in potassium conductance, the more you have inhibition, because potassium moves out of the cell. Then we have this Gabaergic receptors. Gaba A is divided into Gaba A, Gaba B, and Gaba C. Gaba A and Gaba C are both ionotrophic receptors, and they increase the conductance of chloride ion. Chloride ion is going to enter the cell and is going to cause the reduction of the membrane potential. That reduction in membrane potential will cause inactivation or inhibition of the cell. That's why Gaba A and Gaba C are inhibitory potentials. Then we have Gaba B. Gaba B is a metabotrophic receptor. It acts by increasing the potassium ion channel and reducing calcium ion channel via the CAMP receptor. So this Gaba B is also an inhibitory mechanism, inhibitory receptor. So the next one, we have glutamate. Glutamate, we have AMPA, we have carnate, we have NMD, we have N-glycerol. These three are all ionotrophic receptor. M-glu is metabotrophic receptor. Since it increases these ions, so it increases all these ions, you see that it is going to be much more activatory with respect compared to this one here. So now we have glycine. Glycine is going to be an ionotrophic receptor. It increases chloride ion inside the cell. So since it increases chloride ion, implies it's going to be inhibitory. We have histamine, H1 and H2. Both of them are going to be metabotrophic, but H1 is going to act via a secondary messenger called IP3 and DHG, and H2 occur via CMP, secondary messenger. Then we have neurotensin, which is going to occur via the receptor, which is metabotrophic, and then it's going to, this metabotrophic receptor is inhibitory. It both reduces the CMP, inhibits CMP, and increase IP3. Then we have again norepinephrine and epinephrine. So we need to know that the norepinephrine and epinephrine act on the adrenergic receptors. Adrenergic receptors, we have alpha 1 receptors, we have alpha 2 receptor, and this alpha 1 receptors can go from A to D. I also have alpha 2 receptor, we can go from A to C, and we have beta 1 to 3 receptors. Now this alpha 1 receptor, all of them are going to be metabotrophic, and they increase the potassium ion concentration inside the cell. Since they increase potassium ion movement conductance, so it implies that what? It implies that they are mostly going to be inhibitory. So this is why alpha 1 adrenergic receptor are mostly inhibitory, and they act via the secondary messenger IP3 and DHG. So we have alpha 2 receptors. Alpha 2 receptors also are metabotrophic, acting by inhibition of the CMP, but now this time they also increase the potassium ion channel, and they reduce the chloride ion, they reduce the calcium ion channel, so they are also going to be mostly inhibitory. And lastly, we have the beta 1 to 3 receptor, which are also metabotrophic, and they are going to increase the CMP level. They are mostly activatory. So now let's go to the neuropeptides Y. We have Y1 to Y2. So all these are the other ones involved. So the main ones have already been said. So you can visualize now the rest by pausing the video and knowing what they are involved in. So now from here we'll say thanks for your kind attention, and let's go to the next tutorial called muscle physiology. Thank you. Please don't forget to like and subscribe to the channel. Thank you.